We're pleased to be hosting tonight's presentation, The 5 Biotypes of Depression and Advanced Nutrient Therapies, with Dr. William Walsh. William Walsh, PhD, is president of the nonprofit Walsh Research Institute and a key scientist driving the development of nutrient-based psychiatry. His book, Nutrient Power, is the result of his more than 30 years of research and clinical experience. It describes his advanced nutrient therapies for autism, behavioral and learning disorders, depression, schizophrenia, and Alzheimer's disease. His book is available for sale in the lobby, and he will sign copies of it as well. Dr. Walsh has authored more than 200 scientific articles and reports and holds 5 patents. He also directs an international physician training program in the United States, Australia, and Norway, and has developed biochemical therapies that are used by doctors throughout the world. He's a member of the Professional Advisory Board of the American Nutrition Association. As I mentioned, we're very, very proud to have him. He's graciously agreed to take questions at the end of the presentation. And again, it's just a thrill to have a scientist such as Dr. Walsh on our Professional Advisory Board. I mean, it speaks to the quality of people that we have speak at this organization and associated with this organization. And in terms of someone transforming lives of people who are very ill, but who can be seriously and radically transformed through nutrition and nutrients, you will not find somebody who has done more in that realm. And so it's an absolute honor and a pleasure and a treat to have Dr. William Walsh with us this evening. Please join me in welcoming Dr. Walsh.
Got to put it way down. I wasn't as blessed as he was. I was more vertically challenged. Well, it's good to be here tonight with a group that used to be NOAA and now is ANA. And I'm going to be talking primarily about depression. And one of the reasons is in May, myself and my colleague, Dr. Robert DeVito, a psychiatrist that many of you may know, we've been working on this for a few years, and we thought that the world of psychiatry, mainstream psychiatry, needed to know a lot more about depression and better ways to treat it. And they invited me to come to the annual meeting in New York City last May, and so I'm going to be telling you sort of what I told them, and that'll be a major part of what I'll be talking about. So my main topic is the 5 biotypes of depression and advanced nutrient therapies. Now, I think I've learned more in the last 6 years about the brain and about how to help people with serious problems than I had learned in the previous 25 years. And a lot of it has to do with new understandings about the emerging field of epigenetics and new understandings about methylation. How many people here have never heard of epigenetics? A couple of you. Well, before the evening's over, you'll know a bit more, but you will know some part of it. It's something that everybody is going to know a lot about within 5 years because it has so much to do. Sue is waving at me, which I think means that
just the microphone should be more close.
I need to get closer to the microphone.
Yeah.
Okay, is that better?
Can you hear?
Maybe I'll hold it.
No, yeah, so we need to.
How's this going?
Very good.
This will work better. My organization as ANA is also a not-for-profit public charity, a 501c3. Our expertise is really in brain science and treatment, evaluation and treatment of behavior disorders, ADHD, autism, depression, anxiety, bipolar disorder, schizophrenia, and most recently, Alzheimer's. We have an international physician training program that is just growing rather rapidly. We just today got invited to return to Norway for training doctors there. We've been to places like Ireland. I've been to Australia several times, and most recently in the USA. And we believe that by the time this calendar year is over, we will have trained 150 doctors. Our goal is to train 1,000 doctors within 5 years, and I think we're well on the way. And research, I have 4 different research projects currently underway. One of them is completed, and I'm trying to get the final draft of it completed. We were the first people to ever measure metal levels in brain tissues of autistics compared to controls. And there's some very surprising and very intriguing results we found. We had the first data the world's ever seen with respect to that kind of chemical analyses for autism. And we have a project on schizophrenia that we're about to start with some Australian colleagues. We have a behavior study that's just started with Griffith University in Australia studying our treatment efficacy for people with children with violent behavior. Now, I've always been a number person. I started as a scientist. I worked at places like Los Alamos Institute for Atomic Research in Iowa, Argonne National Laboratory for 18 years. And as I got involved with mental illness conditions, I always collected a lot of data. And it turns out that I now have, I believe, the world's biggest collection of chemistry data for a number of different conditions. We have a massive chemistry database based on the 30,000 people that we've evaluated. And it's not just that we have more than 3 million chemical test results for patients with these disorders, but we also have a collection of their symptoms and their traits and their medical histories. And putting all that together is really a fountain of great information. So just to summarize it all, what have we learned from all of this chemistry data and all this information? And basically, it doesn't take a genius to look at this information and find out that there are striking blood and urine chemistry differences between, in this case, the depression population and the rest of society. The same is true of autism. Their chemistries are even more severe and more prominent than depression, schizophrenia, behavior disorders, ADHD, bipolar disorder, anxiety. In every case, there are striking differences between them and the rest of the population. So what are these imbalances? This is one of the most fortunate things that we encountered. First of all, there are more than 300 nutrient factors in the body that are really important to good functioning. However, what I've learned over the years is that there are really only 6, 7, or 8 nutrients that are dominant in brain function and dominant in mental health. And that's really convenient, because if we had to do lab testing for 300 different factors, that would be really difficult to do. And if we had to correct chemical imbalances of dozens and dozens and dozens of factors, it would be difficult. So fortunately, these are the primary factors that dominate mental functioning with respect to things like depression and anxiety, methylation disorders. Undermethylation and overmethylation are important factors. Zinc deficiency we see probably more than any other chemical imbalance across the board with these different kinds of conditions. Folate deficiency and folate overload. We just learned recently why folate is so dominant in so many mental illnesses. And it has everything to do with epigenetics, because folate is one of the chemicals that has so much to do with gene regulation. And that means a lot to do with what happens at brain synapses. Pyrrole disorder, toxic metal overload, and then fatty acids, especially EPA, DHA, and arachidonic acid. Deficiencies of any of those three can cause major problems, because there are more than 100 fatty acids in the human body, but there's only 4 of them in high concentration right at the synapses of brain cells. And these are the 3 most important ones. I think DHA seems to be the one that's most dominant. Anyway, these particular factors have a really powerful effect on mental functioning. And if one could just simply correct these particular factors, most of the patients that we see would be dramatically better. Can everybody hear me in the back? Raise your hand if you can. Now, I get a lot of calls from doctors, and I have frequent questions from mainstream medicine doctors and even researchers. And what they'll often say when they hear reports that we have treated thousands and thousands of patients, and we have thousands of reports of people who've done well, and that we've published peer-reviewed journal articles showing efficacy, A lot of them will call wondering, how did we do this, and they want to know about the biochemistry. And so when they call, I will talk a lot about the biochemistry and brain function, and at some point, they'll ask the classic question, well, what did you do? What was your treatment? And my answer is that the weapons we use are nutrients, amino acids, vitamins, and minerals. And so at that point, I lose about 2/3 of them. But about a third of them stay on the line, and I keep hearing the same question. How could vitamins and minerals possibly help a patient with a serious mental illness? Don't you really need a powerful drug to get the job done? I hear that most often, I guess, for bipolar and schizophrenia patients. Well, nutrients have great power, and the great power resides in these 4 primary areas. First of all, we don't have a lifetime supply of neurotransmitters, serotonin, dopamine, et cetera. There's more than 100 neurotransmitters, but we don't have a lifetime supply of them. They are created, they're synthesized all day, every day in our brains, especially the most important ones like serotonin and dopamine and norepinephrine. And the only raw materials for these neurotransmitters, these really vital, important brain chemicals, are nutrients. Okay, the second is the part that we have learned most recently, and that nutrients have a powerful effect on epigenetic regulation of gene expression. That's what epigenetics means. It has to do with the regulation of our genes. We have 23,000 genes, and every gene's only got one job, and that job is to make one particular protein. And if that protein enables a reaction to go, it is an enzyme. And we now, with the knowledge of epigenetics, we now have the ability to do something we've never been able to do before in nutrient therapy. And that is, we now have the ability to regulate enzymes. We never knew how to do that before, genetically expressed enzymes. The third issue is that we now have the ability that's growing all the time to directly affect reuptake processes at synapses, and that's especially important, I think, in depression. We now know that SSRIs are serotonin reuptake inhibitors. Well, now we know what nutrients can do the exact same thing without side effects. We also know what nutrients can make reuptake worse. And so it's giving us new information, especially, again, in the last several years, that is sort of guiding our treatments and helping us do a better job of helping people. And then the fourth area is antioxidant protection. Virtually every condition that I showed before, whether it's schizophrenia, behavior disorder, ADHD, or autism, or whatever, they all have one thing in common. They all have an issue with elevated oxidative stress. They all have oxidative stress, and very often to an extreme degree. And that seems to be part and parcel of almost every form of mental illness. But nutrients are able to combat every one of these areas, and this is where the power of nutrients reside with respect to brain function. Again, let's go to the first one. That's the synthesis of neurotransmitters. As I said, the brain is a chemical factory, and these are synthesized all day, every day in your brain. And the raw materials are nutrients, and if you have a genetic or epigenetic imbalance, if you have the wrong, radically wrong levels of these nutrients needed for synthesis of a neurotransmitter, you could expect there'll be something wrong with your brain function. And just to give a few examples, and this again is synthesis. This is, I don't know if you can see that in the back, but this, I have a pointer here. I think I have a pointer. Not sure how to make the pointer work. Anyway, this is the final step in the brain of serotonin synthesis. This is where our serotonin comes from. And of course, low serotonin has been associated with clinical depression for many years. Well, it comes from 5-HTP, from 5-hydroxytryptophan, and it converts to serotonin by this particular reaction. And if you notice on the bottom, it lists PLP, also known as P5P, pyridoxal 5-phosphate, which is a form of vitamin B6. Vitamin B6 is a very necessary and important cofactor in this reaction. Well, there are many people who have rather striking deficiencies of vitamin B6, and they tend to be low serotonin people. And yes, you could probably help them with a antidepressant medication, but you also could help many of these people by simply normalizing their B6 levels. And another example is norepinephrine. All your norepinephrine comes from dopamine. And this reaction depends on copper. Copper is a really dominant factor in this synthesis. And it's copper plus plus, divalent copper. And now, the body has a remarkable ability for healthy people to have copper levels that are homeostatically controlled. In other words, you could be chewing on copper bars all day, and if that system's working, your blood copper level will still be okay. However, a lot of people don't have that ability to regulate copper. And for example, if copper were to suddenly elevate, what happens? And it happens to a lot of people. For example, people with postpartum depression, almost all of them are people with extraordinarily high copper levels. It results in lower levels of dopamine and higher levels of norepinephrine. Dopamine is sort of a feel-good neurotransmitter. It has a lot to do with again, feeling good, but also with cognition and mental functioning. Norepinephrine, if that gets elevated, that's a recipe for anxiety and sleep problems, and in some cases, psychosis. Another example is dopamine itself. Dopamine comes from L-dopa. This is the last step. In this case, B6 is very important in the development of proper amounts of dopamine in your brain. And the last example is GABA. GABA, we know, is it is a neurotransmitter. It's one of the two primary neurotransmitters in the brain, the other one being glutamate. And as far as having the most receptors and the most activity going on. And again, you need B6 in order for this reaction to go, but you also need zinc to regulate your GABA levels. So that's just an example of 4 of these cases that show the importance of specific nutrients with respect to the synthesis of neurotransmitters. How many people here have heard of pyrrole disorder? About a fourth of you. Pyrrole disorder is something that was discovered jointly by Abram Hoffer and Carl Pfeiffer many years ago. And what they found was that people that had this particular problem, they discovered it by noticing that they were working on a study of schizophrenia, and they found that some of the schizophrenics' urine changed color while they were storing it in the refrigerator waiting to go to the lab. It turned into a sort of a mauve, purplish blue color. And then they wondered, well, gee, I wonder if that means anything. And then they did a survey, questioned the people who did and did not have, the schizophrenics who did and did not have that. And eventually they found it had to do with something called a pyrrole molecule. And they called it cryptopyrrole. We now know it's a different molecule, but it is a pyrrole. And people who have some people have elevated levels of pyrroles. We all have pyrroles in our body. Basically, they're byproducts of biochemical reactions that are important that go on in your spleen and in your bone marrow. But if you have too much of it, it's a problem because your body just clears this out of your body. It goes through your bloodstream, through your kidneys, and out in the urine. The problem is the pyrrole molecule has a great affinity for any kind of a chemical that is an aldehyde, for those of you who are chemistry people. And the number one aldehyde in the body is B6. B6, and so this disorder strips B6 out of the body. And I just showed you 3 neurotransmitters that are dependent on B6. Well, people with this disorder have extraordinarily low levels of B6. The pyrroles also have the ability to strip zinc out of your body, and that's a double whammy. Those are 2 very important nutrients with respect to brain function. And so if you have this, you would tend to have reduced serotonin, dopamine, and GABA. You would have depletion of glutathione, metallothionein, cysteine, SOD, and catalase. This is just a short list of some of the major antioxidants. If you have pyrrole disorder, you will have an antioxidant problem that's severe. And anyway, supplements of B6 and zinc can usually normalize these levels, often within just a couple of weeks, and often resulting in elimination of symptoms and elimination of the need of psychiatric medication. We first discovered how well people respond to this therapy with violent children. We had seen eventually more than 10,000 cases of behavior disordered children and adults. And if we found they had pyrrole disorder, if they flunked the urine test for pyrrole disorder, we would rejoice because we thought this is going to really work, or is likely to. And we had reports from many families that very violent, aggressive children became quite normal within just a few days, really. It's the fastest imbalance to correct. Now, antioxidants. I'm sure the people in this group know a lot about antioxidants, and I think antioxidants are something that the world needs to know even more about, and especially as people get older. Oxidative stress is pretty clearly involved in dementia and aging, but people throughout their entire lives need to have a proper amount of antioxidant protection. Now, we all have, if we're healthy, if things are working well, we all have remarkable antioxidant protection. Glutathione is the first line of defense, and that comes from the methylation cycle. Your glutathione comes from diet, and it's wonderful the way it works. It's wonderful at combating oxidative stress and eliminating the problem with it. Sort of disables it in a sense. Problem is it's got a limited supply, and it's dependent on diet, and it depends on how well your bodily processes are working. So the backup system is metallothionein. That is a protein that is genetically expressed. It's a zinc-based protein, and that's the backup system. So if if you have a lot of oxidative stress, or if you say you have a serious injury or some serious infection, you're going to run out of glutathione, and then metallothionein takes over, and they work together. They work as a team. In fact, when the glutathione is, you might say, used up and becomes oxidized, it's the oxidized glutathione that turns on metallothionein. It's a very neat system how they work hand in hand. And then we've learned that selenium is necessary for both of them and increases the kinetics of this by 50%. So these three, I call them the three musketeers of antioxidant protection. And of course, there's many other antioxidants, vitamin C, vitamin E, on and on. So now I want to get to the subject of methylation. Again, we are learning more and more now about epigenetics, and it's a really important role in mental functioning. And methyl is the dominant factor. And when I say methyl, it's really acetylmethionine, which is the methyl donor in the body that comes from dietary methionine. And the methyl-folate ratio has a really powerful impact on neurotransmitter activity at synapses. It's the neurotransmitter activity that counts, not so much the amount of serotonin you've got, but the number of interactions, the number of messages that it's sending to other brain cells. Turns out that more than 60% of anxiety and depression and psychosis patients exhibit a serious methylation imbalance. Now we've studied methylation in more than 30,000 people. And about 70% of human beings, as far as we can tell, have normal methylation and don't have that as a factor. 22% are undermethylated, 8% are overmethylated, and they have completely different issues, problems, and neurotransmitter abnormalities if you happen to have methylation disorder. There are two types of methylation problems, and as I said, undermethylation and overmethylation. Now, there's been a lot of interest in nutritional groups throughout the world, really, in undermethylation, and people are now doing genetic tests, the 23andMe and other tests, to see if they have a MTHFR or some other kind of what's known as a SNP, a single nucleotide polymorphism. It's a mouthful to say, so they call them SNPs. But SNPs are basically mutations. Everybody here can be pretty sure that you have probably at least 1,000 or 2,000 SNPs, mutations, and that's why we're different. That's why we're different. That's why one person has green eyes, the other one has blue eyes. 99.9% of our DNA is pretty much identical. It's a 0.1% that's different. And these are mutations that have developed over the years. I think cavemen used to be a lot more similar to each other than we are now. So what is the incidence of undermethylation? Again, this is 22% of the general population. Autism spectrum, 98%. I think sometimes it might be 100% if the diagnosis was more accurate. I'm proud to say that it was our chemistry database that discovered this back in 1999. We were the first ones to discover that autism involved undermethylation. And now there's an army of really first-rate scientists that are studying methylation related to autism. And there are therapies now for autistics that involve coping with the effects of this undermethylation. I used to work with a lot of criminals. I've studied more than 800 felons, including a number of serious cases of, we've studied people like Charles Manson, Richard Speck, and 800 people who've been convicted of serious crimes. So I got to know a lot of people you would call sociopaths or antisocial personality. Virtually all of them are undermethylated. Whoops, I've gone too far, but I could go back. Here we go. Sorry about that. So 95% have antisocial personality disorder, and part of being that severely undermethylated means they're strong-willed. If they commit a crime, they tend to do it the same way, they have the same MO or modus operandi, and they're born with it. Sociopaths are not created by their environment, but they're born with this. Schizoaffective disorder. 90%. If we find a patient who has a diagnosis of schizoaffective disorder and we think the psychiatrist was good at diagnosis, we'd be pretty sure they're undermethylated, and they would flourish on treatments related to that. Oppositional defiant disorder, 85%. Anorexia, 82%. Most anorexics and bulimics are undermethylated and respond very nicely to methylation therapies together with counseling. That's a great combination for permanently correcting those disorders. And then depression itself, 38% of all depressives are undermethylated. Mainstream medicine behaves as though 100% of them are. Okay, overmethylation. Again, 8% of the general population are overmethylated. So you get about 3 undermethylated people for every overmethylated. This is really high in incidence in panic and anxiety attacks. Overmethylation means, among other things, really high norepinephrine levels and high adrenaline levels. So these people tend to be hyperactive, they tend to be moving around and are prone to extraordinary anxiety. And in severe cases, it can be the primary imbalance in schizophrenia, especially paranoid schizophrenia. ADHD, I did it again. ADHD is 28%. That's a pretty high number, really. In other words, overmethylation is the dominant problem for more than 1/4 of all people diagnosed with ADHD. 23% in behavior disorders, depression, 18%. And I'm going to focus on depression in a few minutes. So what's the cause of it? Why are some people undermethylated and while others are not? The number one cause are mutations, these SNPs I mentioned, enzyme mutations. Enzymes, again, from genes that are expressing, genetic expression of these enzymes. And if you have a SNP is a single nucleotide polymorphism. And I'm sorry if I'm making some of you fall asleep here with technology. I'll do my best not to linger on this. But we now know that there are enzymes that are weakened by these mutations. And in the methylation cycle itself, which I'll show you in a second, the MTHFR, we know, is the number one, it's the rate limiting step in this complex cycle I'll be showing you in a second. But we know that if you have an MTHFR SNP, and there's 2 major types of that will weaken, not destroy or disable, but it will weaken the functioning of that enzyme. MTHFR, for example, is a huge molecule. It's got more than 500 amino acids in it. Its molecular weight is 77,000. It's just a really big one. In a single nucleotide polymorphism, what that means is that out of those 500 amino acids, one of them is in the wrong place. You might have arginine where methionine might be, or something like that. That's all that a SNP is, and that's why we all have so many of them. In fact, people have now looked at the genome and studied lots of people. They've now identified more than 10 million SNPs in the human population. So if you do a test and somebody says, aha, you've got a you have a SNP, don't be surprised. You have lots and lots of them. Most SNPs don't have any effect on a person. There are some people that are greatly affected by MTHFR and other SNPs. Others are not affected at all. So, histamine overload can also occur. There are some biochemical, some people tend to have really high histamine levels, and histamine and methylation tend to be opposite because methyl is the way that histamine is metabolized or destroyed or regulated. So some people have it for that reason. And then you could have protein deficiency because you need protein, you need methionine in your protein to give you the methyl capability in the first place. And then, of course, malabsorption. If you're not able to get the amino acids into your bloodstream functioning, you'll be low in methyl. So those are the primary causes, but number one are these enzyme SNPs. So here's a cycle. I'm not going to put you all to sleep by going through the methylation cycle. How many people here are very familiar with this cycle? Probably only 7 of you. Okay. I'm not going to put you to sleep, but again, if you look at the top, I'm looking for a pointer that doesn't seem to work. No. I should have paid more attention when somebody was telling me how to do it. But it all comes from methionine. At the top chemical is methionine, and that comes from our diet. And then it goes clockwise. The methionine converts to SAMe, S-A-M with a little E, and that's easier to say than S-adenosylmethionine. Well, that is the major methyl donor in the human body for many, many really important reactions. And if you don't have enough SAMe, you're undermethylated. If you have a glut of SAMe, you are overmethylated. And basically, SAMe is a unstable molecule that gives up its methyl group really readily. But then this whole cycle is aimed at regulating the amount of SAMe that is produced, and basically it's regenerating methionine in SAMe by going through the cycle. The cycle is, to me, is sort of like the Indianapolis Speedway, and it's like you have vehicles that are just zooming around constantly. There are more than 1 million methylation reactions every second in your body. And so this is going over and over and over and over. And what can block it, what can slow it down, is if the enzymes, now those little green, yellow, greenish ovals are the enzymes. And this is most of the key enzymes, including on the left MTHFR, which is probably the most important of all of them. But if you have a mutation or a weakness in one of those enzymes, then you can become undermethylated because of that. So you can think of this entire methylation cycle as maybe sort of almost like a factory that generates the SAMe or the methylation that your body needs. Now, if you look at the bottom, part of the way you convert when the SAMe gives up its methyl, it becomes something called SAH or S-adenylyl homocysteine. Sorry for the long word. But then that converts to homocysteine. And then part of the homocysteine goes south in that route, and that's where your glutathione comes from, and all these wonderful sulfur antioxidants. But and then the homocysteine is regenerated back to methionine, and there's 2 different ways that happens. Number one, trimethylglycine can do it directly, and that's that orange line that goes up the middle, again with an enzyme enabling that to happen. And then the other route, the one that seems to be more important in most cases, is what I call the folate regeneration. That's that circle on the left, and that's where MTHFR and a couple of other enzymes are needed to function to make this all work. And so you have enough SAMe, so you have enough methyl. And again, 70% of the population, that works well. So that's undermethylation. That's what can go wrong and cause undermethylation. Well, how about overmethylation? What happens to the SAMe? There are more than 80 really important methylation reactions in the body. And some of them have a lot to do with mental illness and with synaptic activity and brain function. And it turns out that most of your SAMe goes to one single reaction, and that is the synthesis of creatine. Creatine, that's the stuff that helps you build muscles. It's different for every person. Some people, for many people, it's 70% of all the methyl. For some people, it's as low as 40 or 50%. But most of your methyl goes in that direction, with the other 30 or so % for other important methylation reactions. So let's look at creatine synthesis. This is what happens to most of the methyl. Well, there are also enzymes that enable this. So if you've got a weakness in one of these enzymes, especially the one on the left, the AGAT, then what happens is that reaction, the SAMe converting to SAH, a lot of it doesn't get used, and you have too much SAMe. You have a glut of SAMe for these people, and that's what overmethylation is. Primary cause of it appears to be impaired creatine synthesis, SNPs again. You could also have a deficiency of the amino acids that you need to make it, and then impaired. Anyway, there's two other ways that this could also occur. But this is what I really want you to realize: is it's a tug of war. It's a methylation tug of war in methylation, and you have SNPs. You have genetic mutations that are tending toward undermethylation, and those that are tending toward overmethylation. Now, a lot of nutritional practitioners now throughout the world actually are using genetic studies, but they focus on undermethylation, and they say, aha, if you have an MTHFR SNP, especially if it's what they call a 677T SNP, they say, aha, you're undermethylated and you need methylfolate or some folate concoction to help you. And unfortunately, that really is not a good way to do things, and I'll explain why. Because you also have SNPs, these mutations that are tending toward overmethylation, and the question is, who's winning the war? You can't tell from genetic data. Fortunately, there are lab tests for methylation status that do a pretty good job of enabling us to tell methylation status, whether a person's over or under. And then we also have, fortunately, we have symptoms and traits associated with undermethylation and overmethylation. So if you use one or both of these tests, one of them is the SAMe-SAH ratio, recently developed probably 6-7 years ago by Drs. Data right here in Illinois, and then whole blood histamine, which we've been using for more than 30 years. And I just want to say it again, present genetic tests for MTHFR and other SNPs cannot determine the net effect of the SNPs that enhance or depress methylation. They can't determine that net effect, but fortunately we have a couple of lab tests that can tell you who won the war, whether it's over or under. Now, Mike mentioned biochemical individuality, and that's probably the hallmark of everything we do. Every human being is different. Humans exhibit great diversity in blood and brain chemistry. But because of genetics, because of epigenetics, and also diet, most people are deficient in several nutrients and overloaded in other nutrients. Overloaded in some nutrients. It's not all deficiencies. So what are the deficiencies that we see most often that impair brain function? And again, it's a relatively small list. Zinc, methionine, folic acid, B6 and B12, niacin, and then fatty acids, DHA, EPA, AA, antioxidants, and magnesium. Well, it took me a few years before I realized why these particular nutrients kept showing up in so many different disorders. For example, zinc deficiency you see in behavior disorders, you see it in ADHD, you see it in many forms of depression, you certainly see it in autism. And then the same is true of these others. Why are we seeing the same ones over and over in so many completely different disorders? Well, eventually it dawned on me that the reason is that these happen to be the very nutrients that have a powerful effect on either brain synthesis or on neurotransmitter reuptake and neurotransmitter activity. These are the major ones. It's not the others. Well, overloads. Well, here's a short list of overloads. If you have a genetic tendency for copper overload, folic acid overload, iron, methionine, SAMe, or even an inability to keep your toxic levels normalized, you can be pretty sure you're going to have a problem. Probably the most surprising thing for me when I first got involved in clinical practice, say the first 1,000 patients out of the 30,000 we eventually saw, was that nutrient overloads cause more mischief than deficiencies. And that's why multiple vitamins usually won't help, because if we were to take anybody in this room and do a complete metabolic analysis, we would probably find that that person had a few nutrients that they were deficient in that were important, and that life would be better if they only knew which ones they were, and they might need many times the RDA because they're fighting genetics. But then they also need to know which nutrients are in excess of causing mischief. And if you take a multiple vitamin, the odds are you can do as much harm as good. And for example, hyperactive children, we found that most multiple vitamins, no matter what the quality of the vitamin was, of the combination of vitamins and minerals, most of them got worse. And the reason is copper tends toward hyperactivity, and most of these hyperactive kids just got more hyperactive. And eventually we found out why. Well, now I'm going to say a little bit about epigenetics. I promise not to say much about it, but I think everybody needs to know a bit about it because this has so much to do with it. This has to do, we now know that epigenetics is a primary cause of cancer, a primary cause of heart disease, and there's growing evidence that it is the primary problem in schizophrenia and in several other mental disorders. And again, we have more than 20,000 genes in every cell's DNA. You've got 1 trillion cells in your body. 1 trillion, that's a lot of cells. That's roughly equal to the number of trees in the United States. However, in your body, every part of your body needs different chemicals. Well, the chemicals come from, many of these chemicals come from genetic expression. And the problem is you have exactly the same DNA, every part of your body. Your DNA is the same everywhere, but you need your DNA to be making a different menu or a different combination of these proteins and enzymes. Your liver needs different ones than your skin and your eyeballs and your kidneys, et cetera. That's accomplished remarkably before you're born. And at 9 months of gestation, a remarkable process called epigenetics is a system that turns off the proteins you don't want in different areas. And people aren't quite sure how that happens. It happens so well for most people. But that's what epigenetics is. It's a natural process that if it's working well, will provide us with good chemical health every part of your body. So they call and most of it is done by methylation, by methyl groups that attach to certain areas of your DNA, and basically, usually, methyl tends to shut off an unwanted protein that you might need somewhere else, but will shut it off, say, in your liver. But what happens is that environmental insults at any age after birth, for example, can alter gene bookmark and produce an indelible or a permanent mental disorder or some other disease condition. These are environmental insults that can take people who are weakened or are sensitive to these insults. And that's what we think is, for example, we know about regressive autism, right? I've seen 6,500 families with autism. I think we've seen more than anybody in the world. And I keep hearing the same sad story of how these little children were wonderful and they were happy and singing and charming their grandparents when they were maybe 15, 16, 17, 18 months old. And then disaster happened, and usually happened really quickly, that this child suddenly developed this terrible disorder, and usually they lost all speech. They stopped at any attempts to communicate with anyone else, including their mothers. They developed GI tract problems. They developed food sensitivities. Many of them became quite ill. And their family was told that your child is autistic and there's no cure, and he or she will have a permanent severe disability the rest of his life, his or her life. Very sad story. Fortunately, we now know that autism is treatable, recovery is possible. So fortunately, we've now advanced to the point where that can be done, although mainstream medicine doesn't quite agree yet, but they will. But anyway, what could cause such a dramatic change? And one of the mysteries is once autism regression occurs, or once a schizophrenia breakdown occurs, and these can also happen rather quickly. One question is, what on earth is going on in the brain and in the entire body that could possibly cause such a dramatic change in functioning? And like at the molecular level, what on earth is going on? But the second mystery has always been, why doesn't it go away? Why do these conditions tend to persist through the rest of your life? I think the only reasonable answer is epigenetics, And I think that researchers need to dive into epigenetics with respect to these various disorders. And I think that's where effective treatments will come, that's where prevention will come, and very possibly a cure for things like cancer and schizophrenia. Well, so that gives you an idea of how important epigenetics is, and you're going to hear more and more and more about this. DNA methylation, your DNA is pretty remarkable. It's a little tiny strand of individual molecules connected together. And if you were to take the DNA and you have 1,000 cells, a trillion cells, and every one of these tiny cells has got a little tiny nucleus, and in there is jammed together your DNA. If you were to stretch it out, it would be about 6 feet long. And this just illustrates methylation that is, in this case, a gene in this area probably would not express. It would be turned off. There's 2 parts to the epigenetics. One is what happens in the womb, and that's called DNA methylation. That gets all set up before a child is born, and that has a lot to do with whether a person is over-methylated, under-methylated. Some people are talkative, others are kind of quiet, some people are obsessive-compulsive, some people are perfectionistic, others are not, some are athletic, others are not. A lot of that is set up by this epigenetic positioning of these methylation marks on your DNA before you're born. And anybody here who's I met somebody tonight who had 8 children, and she could tell you, I'm sure, that how different they are. And this is why they're so different. But the other, and one of the problems is that illnesses that have to do with DNA methylation are so far we don't know how to treat them. Cancer researchers are making a lot of progress on finding ways to correct these bookmarks, these methyl bookmarks that are not supposed to be there. But basically we don't have a treatment for that yet. The good news is that the second part of epigenetics is called histone modification. Your DNA wraps around, see those little balls on the right-hand side? Those are what are known as histones. Histones are made up of 8 linear proteins that are wrapped together like a ball of yarn. And in your body, your DNA wraps around these histones. And that's one of the ways in which your body can compress and jam all this together into that tiny nucleus. And we are able, with nutrients and with drugs, to modify these histones. We first believed for a long time that the histones were just support structures, because your DNA is a single molecule stuck together and incredibly fragile. And they thought originally that was the only purpose of it. But we've learned now, especially in the last 15 years, that the histones have a lot to do with, they can either inhibit or they can promote gene expression depending on what chemicals attach to these histones. If you notice, this is the histone, and you can see the DNA wraps around about 1.6 times. And this goes on every cell in your body. But you notice these little lines coming out, these linear proteins have what's known as, they call them tails, that stick out of this ball. And what they've learned is whatever chemicals react to those histones, those histone tails, can change gene expression. And in some cases can change neurotransmitter reuptake, and can change, and if it's done right, could change a mental disorder into a well-functioning person. Now, a lot of it has to do with competition between 2 chemicals, methyl and acetyl. And these are the primary chemical groups that are trying to attach these histone tails. And if acetyl wins the war, it tends to promote gene expression. If methyl wins the war, it tends to inhibit expression and shut off the production of that particular protein. Nutrient therapy can change the methyl-acetyl ratio and can adjust the production of these enzymes. For example, the enzymes that control serotonin and dopamine neurotransmission rates, the very thing that drugs attempt to do. Now, a gene cannot express unless it uncoils from these histones. It has to be laid bare, and the reason is that large molecules have to get to the DNA strand in order to enable this. They're called RNA polymerase, and that's the molecule that tends to copy the gene. And then what's known as transcription factors that are very large molecules, genetically expressed. And anyway, these large molecules if they can't gain access to that DNA histone, these regions, if they're densely compacted, nothing will happen. The gene will not produce, it'll shut off the production of that particular protein. Now, why do they attach? We now know that it's electrostatic protection or attraction. We know that opposites attract. Well, the DNA itself is a weak acid, ribonucleic acid. The histones are basic. So they have this gentle attachment because of the electrostatic connection. But if acetyl groups attach to the histones, what happens is you decrease the pH. You decrease the pH, and that means you decrease the attraction, and the DNA uncoils like magic. It uncoils, becomes available then, and you have these large molecules like RNA polymerase. They're just swimming around in the liquid in your nuclei, in the little tiny nuclei, trying to find a gene to express. And that's how it all works. And methylation tends to increase the pH, makes the attraction stronger, and all jams together. And so this is a case of undermethylation. Undermethylation, and as you notice, the DNA strand between the histones is now laid bare, and genes in that area would tend to express. And this is the opposite. If methylation wins the war and you have not much acetyl and you have a lot of methyl, it all jams together, and that shuts off the production of that particular gene's protein, because the important large molecules just can't get there. Now this has a lot to do with reuptake, neurotransmitter reuptake. Back in 1968, maybe up until maybe 1985, we used to think that the important thing in brain function in terms of depression and anxiety and mental illnesses of all kinds had to do with the amount of neurotransmitters. How much serotonin do you have? And there were, in fact, the first drugs were aimed at increasing, first antidepressant drugs were aimed at increasing serotonin levels, like MAO inhibitors. They were designed, and they do, to some degree, increase the amount of serotonin in the synapse. Well, eventually, in the middle 1980s, we learned that the dominant process is not how much neurotransmitter you got. The dominant process is reuptake, which somehow got erased from my slide. I apologize for that. The dominant process is reuptake, and it's far more important than the amount of neurotransmitter present. So reuptake is, in terms of nutrient therapy or drug therapy, that is the name of the game. And what do SSRIs do? They inhibit reuptake. And so that traps the neurotransmitter in the synapse for a longer period. And this is a picture that the top cell might be what you call a presynaptic neuron. That's a brain cell, and when it fires, it shoots out, it sprays out serotonin molecules. But one amazing thing is that when that happens, these molecules tend to quickly go back to that original cell. And they go back through little passageways in the membrane, and these passageways are called transporters or transport proteins. They're proteins that snake in and out of the membrane, and it's amazing how fast that can happen. There are brain cells that can fire every second. So they have to not only shoot out the neurotransmitter, it has to go back really fast. And it's all done by electrostatic mechanisms. And of course, what you see in the bottom is a brain cell, or maybe possibly a receptor, maybe a dendrite or a spine with a receptor on the end. And of course, the neurotransmitter may or may not attach to one of these receptors and cause that cell to fire. And that's really how brain cells communicate. So these reuptake transport proteins are really important. And this is all fairly new. I think when I talked to this group 2 or 3 years ago, I don't think we knew all of this. I'm sure we didn't know all of this. This is a primary determinant of neurotransmitter activity more than anything else, especially at serotonin and dopamine receptors. Concentrations of these are less important, and I mentioned they're transmembrane proteins that I say they remove the neurotransmitters and send them back where they came from. It's like a vacuum cleaner inhaling dust particles. It's amazingly fast. And they are formed by gene expression. And nutrient therapy, we now know nutrient therapy can regulate the production of these transporters. And therefore, we can change the population of these transport proteins, these passageways for the returning neurotransmitters. And that can have a really dramatic, powerful effect on the activities of important neurotransmitters. So what are some of the insights? We're just beginning to learn all of this, but we already know a lot that can already be used to help people. We now know that niacinamide, and acetyl homocysteine, and other chemicals, we know that they are dopamine reuptake promoters. The great Abram Hoffer in the 1950s discovered that he was the first person to discover that nutrients can have a dramatic effect on mental functioning. He found that niacin could really help a lot of schizophrenics. And he had a theory for why niacin was affecting this, and he called it the adrenochrome theory. We now know this is the reason. I wish Abram was still alive because he would love to know this. We know that basically niacin in the form of niacinamide is it's something called a deacetylase inhibitor, but what that means is that it tends to put more acetyl than methyl on a gene. And many people with schizophrenia, probably a third of them have really high dopamine activity, and that's why they're schizophrenic. And that's why niacin was so helpful for these people. Methionine and SAMe, which is now available in every drugstore in America, over the counter. These are serotonin reuptake inhibitors. They do the exact same thing that drugs do, only they do them more slowly. The drugs quickly, within a day or two, can get to these transport proteins. They interact. That's what SSRIs do. They go straight to these transport proteins, and they block the return of the serotonin, and that's why they work. And by doing that, they increase serotonin activity, and some of the depressives have that problem. Folates are really interesting. Folates are very powerful in epigenetics and in mental functioning, and this is one of the hardest things for even professional nutritionists and nutritional doctors to understand. They reduce synaptic activity at serotonin and dopamine and norepinephrine receptors. Even if they're undermethylated people who have low levels of serotonin activity, and folates are wonderful methylators, still the epigenetic process overwhelms that. So we're learning a lot about this, and we're going to know more and more and more. And I really believe that as brain science advances, the need for psychiatric medications will gradually fade away. Folates reduce neurotransmitter activity. I want to highlight this because this is something that most nutritionists get wrong. Folic acid, folinic acid, L-methylfolate, these are all very effective methylating agents. However, folates increase gene expression of these transport proteins. In this case, serotonin transport proteins are called SERT, and that results in reduced serotonin neurotransmission. And if you've got low serotonin depression, that's the last thing you want. And so basically, what's interesting is that methyl and folate are both methylating agents, but they have completely opposite effects on mental health. And most undermethylated depressives with low serotonin activity are intolerant to folates. And we've seen that hundreds of times with patients. Back in May, my colleague and I, Dr. Robert DeVito, who was once Director of Mental Health for the state of Illinois, and was chief of psychiatry at Loyola and trained more than a third of all the psychiatrists and psychologists in the area. He and I have been working on this for several years, and we decided that the mainstream psychiatry needed to know more about better ways to help people with depression. So we were invited to do a paper, and I delivered this in May of this year in New York City. And 17,000 psychiatrists from all over the world at this one meeting. It comes once a year. We have this really large depression database. I think it's the largest in the world. We evaluated 2,000 chemical database, 2,800 patients diagnosed with clinical depression. nearly 250,000 assays of blood and urine for these people. And of course, we compared it with people who didn't have depression. And 320,000 medical history factors. Well, mainstream psychiatry has a misconception. This is a slide I put out at the APA, and I thought they might throw red tomatoes at me or boo, but they all sort of sat and wrapped attention. Depression is regarded as a single entity. Anywhere in the world, if somebody's diagnosed with clinical depression, somebody's going to want to give them an antidepressant medication, whether it's Prozac or Paxil or Serotonin or there's about 12 of them now. And it all comes from a central belief. And by the way, I had hoped that DSM-5, when it came out, would maybe show some of this, but no, it's still all there. The central belief is that depression involves low activity at serotonin receptors, and that the treatment of choice is, at least in the beginning, of course, is SSRI antidepressants to elevate serotonin activity at synapses. SSRIs can do that. Well, from my database, we have found pretty clearly that depression, clinical depression, is an umbrella term used to describe a number of completely different disorders. And we find that there are 5 high-incidence depression biotypes, and these amount to about 95% of all the depressives that we've seen are in one of these 5. And the biotypes appear to be distinctly different disorders, each with unique neurotransmitter imbalances and symptoms, and a separate treatment approach may be optimal for each one of these biotypes. The slides I'm showing you now were some of them I actually gave at the APA meeting. And this is a chart that sort of shows what we have found. These are the 5 depression biotypes. I'll just go through them quickly. On the left side, you see in the blue, 38% of the depressed people that we've seen are undermethylated, and part of undermethylation means low serotonin activity. And these are people who usually reported that antidepressant drugs help them, although sometimes with intolerable side effects. The lower right, the group in purple, the 15%, these are people with severe pyrrole disorder, and they have severe B6 deficiency. And I showed you a graph, a slide before that showed how B6 is needed for making serotonin, and these people tend to have dramatic serotonin deficiency. So these are the 2 groups. It amounts to 53% of the population of depressives, and these are the ones who actually have a good chance of being helped by an SSRI antidepressant. Well, we also have the green on the right, a very interesting group. This is 17% of all depressives. These are almost all females. And these are people who are depressed primarily because of a metal metabolism disorder. These are people with really high, unregulated, very high levels of copper. And as I showed you before, if the person has high levels of copper, that's not supposed to happen because it's supposed to be controlled, but if somebody is unable to regulate their copper levels, that tends to give them really low levels of dopamine, high levels of norepinephrine. Very common in ADHD because Ritalin and Adderall tend to elevate dopamine. Well, these people tend to be, many of the, I think 68% of all ADHD patients we've seen had this problem at copper, but also depressives. It's, I believe, responsible for 90% of cases of postpartum depression and postpartum psychosis. Because during a pregnancy, a woman's copper level more than doubles, and the growing little baby needs that because copper is needed for angiogenesis, for the ability to rapidly create new blood vessels. Now, after the baby's born, that level that is now more than twice of normal is supposed to start heading right back down to normal. For some women, that doesn't happen. They don't have the chemical mechanism for that to occur. Their copper level stays high, and that's anxiety, depression, and in some cases, postpartum psychosis. So that's a 3rd biotype. Now, on the very bottom there, in blue, you see 5%, and that is toxic metal depression. Over the years, 1 out of every 20 people we saw, and their major problem was overload of lead or mercury or cadmium, and That number, by the way, has dropped lately, ever since we took lead out of gasoline. I think that level is down now to about 2 or 3%, so that's a good thing that we've done with our environment. The most interesting biotype, well, that's 4 of the biotypes, is the one on the upper right, in red. 20% of all the depressants. And these are people, we call that low folate depression. That's just the name we gave it because it involves a need for folate. And these are the people who have high serotonin activity. And if you give them an SSRI, they will get worse. And we've had hundreds and hundreds of these people through our clinic. And it seems like every one of them says that their doctors put them on one SSRI after the other. And every time, the anxiety and the depression got worse. In many cases, dramatically worse, and they had to stop. So I'll just quickly go through this. I think I'm running out of time. Nobody's left yet, thank you. 38, undermethylated depression, 38% of our depression population. They have high blood histamine, or they might have low SAMI-SDH ratio. These are the blood tests. High incidence of these MTHFR SNPs. And they have excessive gene expression of SERT. These are the transporters, the reuptake proteins, and low activity at serotonin receptors, which is why they're depressed. Low activity at serotonin receptors. And again, these are the ones who have the low methylation, and so they're making too many of these transporters. So the serotonin tends to really quickly go back in the cell, and this is why they're depressed. Symptoms and traits are really interesting. They're very different from other kinds of depression. If you have someone who's depressed and has a history of a very strong will, OCD tendencies, calm exterior but very high inner tension, competitive and perfectionistic, addictive tendencies. If they start to smoke, they might smoke 3 packs a day. If they start to drink, God help them, or drugs, even worse. Seasonal allergies, 75%. They have a high libido. They have a high sex drive. And SSRIs, usually, for these people, usually you get reports of excellent improvement in mood, although maybe with side effects that people don't like. So the nutrient therapy approach is we enhance methylation, suppress acetylation of the DNA in histones. And SAMI and methionine are probably the #1 weapons we use as serotonin reuptake inhibitors. And we avoid folate supplements like poison because we know that will do the exact opposite. And then we use augmenting nutrients. Biotype 2, these are the high copper females, primarily females. I think it's 95% of them are females. And they have elevated norepinephrine, reduced dopamine, high anxiety, tendency for panic. If we get a patient who has panic disorder or anxiety disorders, we first thing we want to do is look at their mental metabolism and see if this is the reason, and often it is. They have a onset during a hormonal event, which could be puberty, it could be childbirth, or it could be the change in life later on. High incidence of postpartum depression, and they're intolerant to estrogen and even intolerant to progesterone creams. To something, then it's not as serious as the intolerance to estrogen. Many of them have ringing in the ears, sensitive skin. They're intolerant to cheap metals. Their husbands don't like it because they always want expensive metals that are kind of pure because they can't tolerate cheap metals. And SSRI antidepressants usually don't do anything. They don't make them worse, don't make them better. Most of them say nothing happened. And many of these women have been on a whole raft of these antidepressants. And again, this is their problem is they have too much copper. This is the slide I showed before. So they have too little dopamine, too much norepinephrine, and that's bad news for your mental health. Biotype 3, these at 15% double deficiency of B6 and zinc if they have pyrrole depression. High oxidative stress, extraordinary oxidative stress, severe mood swings. Many of them have mood swings that go up and down many times the same day, and a lot of them are misdiagnosed as rapid cycle bipolar. Anyway, explosive anger is common, extreme anxiety and fears, poor short-term memory, because you need B6 to remember what you've read. Many of them have had a history of a reading disorder, little or no dream recall, sensitivity to light and noise, abnormal, if they put on weight, they have abnormal fat distribution. And most of them say something good happens if they take SSRIs, but it's usually partially. Again, because they are low serotonin activity people. And again, serotonin, you need B6, and these people tend to be really low in B6. GABA synthesis, again, they're low in zinc, and these people who have the pyrrole disorder are low in zinc, so they have too little GABA, which is a calming neurotransmitter. Nutrient therapy approach is really simple. We just have to normalize B6 and zinc, and very often with depression, it's all over in about a month. And we've had a lot of people, to their great surprise, see their depression suddenly just disappear, and that they're able to get rid of their antidepressant medication. We know that biotin and primrose oil are also tend to be very low in people with this disorder. Toxic metal depression, I'm not going to go through the details, but it's now decreasing population of our depression population, severe oxidative stress, impaired NMDA function. That's a neurotransmitter of the brain that we're learning more and more about. And it has a lot to do with schizophrenia and bipolar and obsessive compulsive disorder and people who tend to be addicted. Absence of trauma or emotional triggers. They feel bad and rotten no matter what's going on in their life. They just constantly feel bad. Unrelenting depression, abdominal distress, metallic taste in the mouth, bad breath, irritability, anger, and very often food sensitivities. If you take an SSRI and these people can get terribly depressed, usually nothing happens, good or bad. That brings us to the most interesting one, and this is low folate depression. 20% of our population, high anxiety and panic tendency, non-competitive in sports or games. They tend to be really nice people who just aren't competitive. If they're playing a game like Monopoly or Bridge, they probably feel bad if the person they're playing lost. I mean, they're just really wonderful people to have as neighbors. They mostly do not have insulin allergies, but they tend to have very high food and chemical sensitivities. For some reason, most of the people we've seen with really high musical or artistic ability were over-methylated, which is the same as low folate. Underachievement is common because they're just not competitive. They don't care if they get the best grades. They're more social people. Most of them have a sleep disorder. They have a low libido, and most of them say that this is what's really important. These people told us over and over and over if they got SSRI antidepressants, things got really worse. And the approach is we want to enhance acetylation. We want to have more and more of these transport proteins to enable we want to suppress methylation and the histones. And anyway, folate and niacinamide promote acetylation of chromatin, and that's the main weapons we use for these people. We want to promote acetylation for them because in their case, they have too much serotonin and too much dopamine, and you have to reduce it. And the way to do that is to cause more transport proteins to populate the membranes of their brain cells. And anyway, we have to strictly avoid SSRIs, methionine, SAMe supplements. And I think I've talked about this before, but SSRI antidepressants, they work really effectively generally for undermethylated and pyrrole disorder people. They're generally ineffective for copper overload and toxic metal depressives, but they can cause suicidal and homicidal ideation in certain low folate patients, especially young males. Now, anybody who buys an SSRI antidepressant, there'll be an insert, and in there, there'll be a warning that some people have a side effect that involves suicidal ideation or homicidal ideation, especially young men. That's been known for a long time. Well, as I told the APA at this meeting, there are inexpensive blood tests that can identify promising and unpromising candidates for these medications. I also told them that nutrient therapy can often eliminate the need for them. Anyway, blood testing and medical history can guide medication selection. I showed this to the psychiatrists because they're still engaged in the drug therapies, and I want them to know that these tests could guide their medication. Do they use a benzodiazepine? Do they use an SSRI or what? An SNRI or what? Anyway, DSM-5 came out, and 3 of our biotypes are right there in DSM-5. What they call disruptive mood dysregulation disorders, high pyrroles. Major depression disorder, I think those are all undermethylated people. And then they have what they call premenstrual depressive disorder. It's one of the major forms of depression according to DSM-5, and those people are the copper overload people, and almost all of them. So I also suggested that this has a lot to do with school shootings. There's been a consistent pattern and say the most serious cases in the last 20 years. There's 50 cases, 50 cases people have studied in the last 20 years of young people, students, who went into a school and tried to kill people or did kill people. And now I've worked with 10,000 cases of depression, and most children, 10,000 cases of people with behavior disorders, including violent, a lot of them were very violent. And what we found is that in most cases they were violent before they were 3, 4, 5 years old. The families, most of them reported there was this problem that came early on. So most behavior disordered people, you see it early. Not with the school shooters. Most of the school shooters had okay behavior during their formative years. They developed anxiety and depression during their teen years. They were treated with SSRI antidepressants. Depression worsened, suicidal ideation began, and disaster followed in these cases. School shooting often followed by suicide. And myself and a lot of my colleagues are convinced that there's a very strong association between SSRI drugs and school violence. And this again is what the SSRI antidepressants do. The problem is they can cause suicidal and homicidal ideation, and we think it's the low folate depressives that they can identify with inexpensive blood tests. I found a lot of the psychiatrists paying rapt attention to this, and a big herd of them rushed up to me after my talk wanting to know how to do this. And we have 15 of them now in our next physician training program next month. So I have a proposal for preventing school shootings, school violence, and some people say we got to get rid of the guns. Well, there's 300 million guns in the United States, and even if people could agree to do it, it would take forever to do that. Others say we need to find a way to make sure that people who are mentally ill or have behavior disorders, make sure that the wrong people don't get the guns. That would again take forever. But I think this would work and work quickly. So I urge them to have mandatory blood testing prior to SSRI treatment for young males, and really for anyone with depression. And then prohibit SSRIs for low folate patients. So in summary, 5 distinct biotypes of depression have been identified. Inexpensive blood testing can assist in diagnosis and treatment, whether it's nutrient therapy or even assist psychiatrists in drug therapy. The emerging science of epigenetics is providing a roadmap for far better and effective nutrient therapies, and very different treatment approaches are needed for each one of these biotypes. I have a book that we wrote. We have a revised and somewhat upgraded version from the hardback copies that we had originally, and this just came out this year, and these are somewhat advanced from that. And we have some of those available outside. And I want to leave you with Pfeiffer's Law, and that is that for every drug that benefits a patient, there is a nutrient that can provide the same benefit. Thank you very much.